Hello, everyone. I'm Larios, an assistant professor of plant ecology at the University of California, Riverside in the Botany and Plant Sciences Department. And today I wanted to tell you a little bit about my research lab. And so in my lab, we leverage greenhouse and field experiments to address three core challenges for ecological restoration. First is determining when to intervene or when to actually take any management actions. Second is how to increase the efficacy of any given action. And lastly, how do we manage for our ecosystems for resilience? And within these three challenges, we work broadly on a core set of objectives. For determining when to intervene, we are often evaluating the impact of stressors such as wildfire and plant invasions on communities, and to ultimately guide decision-making about when to intervene. For increasing efficacy, we often work to identify potential recovery constraints or barriers to recovery for desired species. And also we work with land managers to identify and evaluate management actions such as grazing or soil amendments and the impacts that they have on other management goals. And lastly, we couple field experiments and modeling to identify management actions that can be used to enhance adaptive management strategies in ecosystems. And we have projects across the state of California, which we are very lucky to be able to have so many different field sites. And we have a strong focus primarily in grasslands. But we have been expanding our research program to also evaluate dynamics in Southern California shrublands, particularly in response to wildfire. And today I'm going to be talking about one project in particular in Northern California that we have been carrying out now for a few years at the Sierra Foothills Research and Extension Center, trying to identify the impacts of plant invasions and these little voles on native plant recruitment in these invaded grasslands. And we've been, or at least the lab has historically focused in grasslands because as a system, they've been greatly impacted by environmental change and in particular plant invasions. So I'm really excited to be able to talk to you about these results from the study that I've been conducting with my collaborators, Drs. Lachlan Charles and John Marin. So in these grasslands, in many instances, what we often see are a mosaic of patches dominated by native species or non-native species, which you see outlined here in the California grassland. And if we zoom in, we can see that the native perennial here on the left is still green in late June, while the neighboring slope dominated by annual non-native species has mostly senesced, suggesting that they are utilizing resources differently in this landscape. But distinct boundaries like this one, and sometimes distinct patches in landscapes like the ones you see here. So on the left, you see an area still dominated by wildflowers, which are all native to California grasslands, versus on the photo on the right, you see an area that's mostly dominated by introduced grasses, right? So these different images that we often find on landscapes and patterns on landscapes have really prompted the question of what ecological processes mediate plant invasions. And from ecological theory, we have learned that the abundance of species in a community is likely to be related to some key functional trait that is representative of a main ecological process structuring the community. So here, for example, we have this hypothetical trait on the x-axis, and on the y-axis, we have species abundance. And the orange filled shapes represent native species, where the abundance of species is positively related to this hypothetical trait. An introduced species may be able to establish within a community if it shares similar traits as the resident species and fills an open vacancy or niche in the community. And here we show this as the white shape, as the white triangle. And in these situations, the species is expected to be an invader that might occur at a low abundance and coexist with the resident species and perhaps have minimal impact on the ecosystem as a whole. Now, if there were an introduced species that were able to reach high abundances outside of this trait abundance relationship, here shown as the star, this may be evidence that the introduced species may become a problematic high abundance invader. And in many cases, when that happens, what we see is that initially plant richness decreases with invasion. So here we show that as the loss of all the orange symbols. But over time, when we begin to see establishment of new species into the system, what happens more often than not is that those newly established species are other invaders. And so within land management, managers are often faced with trying to answer the question of what determines the invasibility of an invaded community. And that by answering that question is complicated by the fact that many invasive species often alter ecological processes to benefit themselves. And one mechanism within California grasslands, by which that may occur, is by increased litter layers. And so many species, introduced species, tend to have slower decomposition of their plant material from one year to the next, and that results in this accumulation of litter in the system. And so here you can see that on the right in a dense field of litter that's present in an invaded annual grassland. And on the right, what you see are all the plants trying to grow through that litter layer the following growing season. And this novel environment may select for different traits being important in driving successful establishment. However, it's not clear why this environment would necessarily favor other non-native species versus native species that may have those traits. Furthermore, in many of these invaded systems, other species interactions, such as herbivory, are still happening and may mediate some of these invader impacts. For example, in many grasslands, small mammals such as voles break down litter as they move through a grassland and forage. And you can see their signature runways here in the pictures in the middle and on the right. And these multiple ecological interactions may impact different traits or may impact the same trait, but have either additive or opposing effects on traits, really complicating the situation. So when we're thinking about traits in plant invasions, we have historically focused on traits that would provide insight into competitive interactions between the invader and the resident species. So there has been a big focus on plant traits that are indicative of resource acquisition, such as specific leaf area and height. But in a high litter invaded altered system, a trait like seed mass that is related to seedling stress tolerance might be more indicative of successful establishment. But those traits that make a species a good competitor may also make them more vulnerable to small mammals, as they tend to prefer to eat higher leaf nitrogen, fast-growing leaves, such as those with high specific leaf area and big seeds. However, it's unclear the extent to which the evolutionary history of the species, so whether they're native or non-native, will impact these dynamics. For example, will small mammals preferentially consume native species, providing non-native species with an advantage? So for this study, we wanted to tease apart which plant traits are mediating establishment in an invaded grassland and how these multiple ecological interactions, specifically invader-mediated litter accumulation and small mammal herbivory, might impact trait performance. And secondly, we wanted to ask, how does the provenance of a species, that is, whether it's native or non-native, mediate these dynamics? So we conducted a field experiment in an invaded grassland at the Sierra Foothills Research and Extension Center in Northern California. The grasslands are predominantly dominated by introduced annual grass species. And at this research station, we set up 8 paired rodent exposure and control plots across an oak woodland habitat. The exposures were about 9 meters by 9 meters, a fence that was buried about half a meter below ground and was about a meter above ground, with that metal flashing all the way around to prevent any small mammals from climbing over the fence and getting into the area. The primary small mammal in the system is the California vole, which moves through the grassland and runways, which you can see here in the pictures, and it tends to prefer areas with high litter layers, as litter can provide some cover from aerial predators. Within each of these experimental sites, we set up smaller half-meter by half-meter plots that received either an exotic seed mix or a native seed mix. And those seeds were added to either a plot where the litter layer was still intact or where it was removed. Secondly, we measured the establishment of species from our seed mix and the composition of the background community in our plots. And then lastly, we measured plant traits for the species present within the seed mix and for those in the background plant community. Lastly, we repeated the seed addition in 2 years to account for interannual variation in species responses. We focused on 2 key plant traits, seed mass and leaf nitrogen, that would capture responses to different ecological processes. For example, we predicted that seed mass would be important in mediating which species could establish within a thick litter layer, but might also be impacted by small mammal foraging. And secondly, we predicted that leaf nitrogen might impact small mammal foraging and influence the number of stems that subsequently reproduce. And for the results that I'll be presenting today, the impact of year was very minimal, so we pulled them together. So if we start looking at some of these responses, what we can see is that we first evaluated the probability of recruitment. So for a given species, did it recruit or not, which is shown here on the y-axis. We found that the probability of recruitment increased with seed size, regardless of the origin of the species. And so here we see the origin of the species in different color symbols, where the introduced species are the orange and the gray symbols, and the native species are the blue and the red. And so ultimately what we saw is that the native and non-native large seed species both had a higher probability of establishing, and this wasn't impacted by the litter treatment in any way. So bigger seeds, higher probability of recruitment. When we then sort of continued tracking the species and looked at their actual density of recruitment, what we saw is that the density at which a species recruited was also influenced by seed mass, such that the number of individuals increased with seed mass, and again, this was independent of species origin. When we then followed the established individuals to reproduction, we observed that leaf nitrogen was the important trait that mediated reproduction. And here we found that species with greater leaf nitrogen had fewer reproductive individuals. Again, we didn't find an effect of species origin, but as you might have noticed in this graph and in the last, there's a fair amount of variation between the yellow and gray dots compared to the blue and red. So when we wanted to look at the average density across our experimental treatments, we found that non-native species recruited at higher densities and at a similar density across all of our treatments. So here you can see that by all the points indicated by the letter D, where the exotic species recruited at higher densities across the board. For the native species, we see that they established at lower densities, and we found that litter strongly impacts recruitment, as density was the highest when litter was removed, here indicated by the letter C. We did observe an interacting effect of voles when litter was intact. So interestingly, we observed that when voles are removed and litter is still intact, natives established at the lowest density, here indicated by the letter A on the graph. Native density slightly increased when voles were present and litter was still intact, indicated by the letter B. And when we looked at the mean number of reproductive stems, we found similar patterns where the non-native species had a higher density of reproductive individuals and were not sensitive to herbivory or litter. And the number of reproductive individuals increased for native species when litter was removed. And for reproductive stems, we didn't see an effect of the rodent treatment in this instance. So what does this all mean? So if we go back to our initial questions, we found that seed mass and leaf nitrogen were 2 important traits that mediated a species establishment and reproduction in this invaded grassland. For how multiple ecological interactions might impact trait performance, we observed that litter in herbivory did not impact the relative importance of seed mass or leaf nitrogen. As having higher seed mass increased probability of recruitment regardless of species origin. And similarly for leaf nitrogen, that had higher leaf nitrogen had lower reproductive stems regardless of species origin. We did observe that litter in herbivory impacted native species, but in a nuanced way. So litter strongly reduced native establishment and reproduction, and native establishment was the lowest when voles were absent and litter was intact. And we observed that small mammals slightly mediated the negative effect of plant litter on native species. Lastly, we observed that non-native species perform better for a given trait and were not affected by litter accumulation or herbivory, suggesting that non-native species are likely responding to a different ecological interaction with the net effect that they may more readily establish into this invaded grassland. So for future studies that are looking at trait responses and trying to use plant traits to infer the relative importance of certain ecological interactions, it'll be really important to take evolutionary history into consideration, particularly as our many ecosystems are sort of changing in composition and incorporating many new introduced species. And so observed trait patterns may indicate different ecological processes based on the provenance of the resident community and the newly establishing species. And so trying to account for this evolutionary history will be really important, as it may contribute a novel source of context dependency in trait-based ecological studies. And with that, I wanted to thank all the individuals who helped us in the field and in the lab and for this particular study, and the Sierra Foothills Research and Extension staff for the logistical support, and for my funders. Thank you for tuning in my talk. Please reach out if you have any questions or comments.